Hello everyone, my name is Marcy and today I'll be speaking about behavior and parasitism in a wild baboon population. So to start us off, a little bit of a background. Parasites have wide-ranging detrimental effects on host health and survival, and as we know, host behavior influences parasite burdens and vice versa. Today I'll be presenting two studies that investigated how behavior influences parasite burden in baboons. Specifically, I'll be focusing on grooming behavior, which is a major social activity for many non-human primates and is known to confer both health and social benefits. So the general study area is the Amboseli region of Kenya, and the study species is the yellow baboon. These animals are subjects of long-term individual-based monitoring, and the data collected includes the life history events, social interactions, and biological samples, including stool samples and blood samples. So the first study looked at grooming received and tick load, and here we had 65 baboons that were darted, and we collected whole blood for blood smear preparation, hematocrit analysis, and PCR screening of hemoparasites. We also collected ticks that were later identified and counted. For each of the subjects, we had the listed metadata, but of importance is the count of grooming received 6 months prior to darting. So the first result, we saw that higher grooming counts predict lower tick loads. On the y-axis is the tick load, and the x-axis the counts of grooming received. And what we can see is this decline in tick load with increased grooming. The second result was for tick load and hematocrit, and we saw that higher tick loads predicted lower hematocrit. Again, on the y-axis, we have the residuals of PCV, which is hematocrit, and the x-axis tick load. So this led us to conclude that grooming reduces tick load and improves hematocrit, which is a health index. The second study was on behavior and helminth burden in female baboons. Here, we measured helminth parasite burdens in 745 samples from 122 female baboons. And the data we had included observations of host environments, social behavior, hormone levels, and reproductive indices. We investigated the drivers of helminth infection and the costs of helminth infection. For the drivers of helminth burdens, we looked at processes operating at different levels, inclusive of the population, the group, and host level. We will lay an emphasis on the high social connectedness because this is a measure of bond strength that is calculated from the frequency of grooming received and given. So for our results, the drivers of helminth burdens included several factors, but when we key in on behavior, what we see is this social connectedness index, which is a measure of social bond strength, was associated with helminth burden such that social connectedness decreases helminth burden, at least the SCI to males and also to females. So for the reproductive costs of helminth burden, we saw that high parasite burdens were associated with longer interbirth intervals. And this was true with the total IBI duration, postpartum amenorrhea, cycling, and pregnancy. For the stress hormones and trichuria species burden, we saw that higher egg counts were associated with elevated glucocorticoid concentrations. So the conclusion from this study was that multiple level processes influence helminth burden. Social isolation was associated with higher helminth burden. Helminth burden prolonged interbirth intervals. Helminth burdens are associated with high fecal glucocorticoids. And a special thanks to the listed institutions. I'll take questions. Thank you.